So in your notes, let's go ahead and go to pericardium. We've discussed the two views of the heart. And now with the pericardium, we're talking about a sac. The heart's in a sac. The sac itself has two distinct layers, but there's actually three components to the official pericardium. So the sac has the outer aspect, which is the fibrous pericardium, which is what we actually see in our picture down below. We see a little insert trying to show magnification of the pericardial sac and the pericardium. The fibrous pericardium is indeed the outer aspect. That's dense irregular CT. We talked about that. Then as you go inside the sac, it has a parietal layer, and so that's called parietal pericardium. And then we have that parietal pericardium that extends over the surface of the heart. That little insert actually does a nice job of showing us that. And that's the visceral pericardium, which also has an additional name, the epicardium. Between the parietal and visceral pericardium, we have a space called the pericardial cavity. So space between parietal and visceral pericardium. It actually has a small amount of fluid in that space, and it's serous fluid. So this small amount of fluid is produced by the serosal surfaces. And we've encountered this before when we talked about our pleural cavities and our abdominal pelvic cavities. So there is this space, and it's more considered a potential space, but whenever there's something in it, that space does exist. A little bit of serous fluid is there. Serous fluid is clear fluid, and it is there for a specific reason. Because the heart moves. When the heart beats, it actually moves or writhes, so to speak, in that pericardial sac. And so in order to minimize friction between the visceral and parietal surfaces, we actually have that serous fluid there. So serous fluid reduces friction. Between the two serosal surfaces, which is your parietal and visceral pericardium. Now, if for some reason there was a bleed into this space, into this pericardial cavity, what would be a potential problem for that? We have blood actually filling it up and tensing it, just like if we had water in a balloon. What would that do to the effective action or movement of the heart muscle itself in that space? It would limit that. That actually has a clinical term, and one day you may encounter that. It's called pericardial tamponade. And that's literally, that fluid is squeezing the heart muscle, causing it not to be able to distend or fill properly, and therefore eject its blood properly. So that's a really bad situation. There's another situation that can actually occur in this pericardial cavity, and it's an itis. So you can actually have inflammation of those serosal surfaces, and when you have inflammation of those serosal surfaces, they stick together and produces that condition called pericarditis. Pericarditis will not cause a big effect on the heart muscle's movement, but because we now have sticky surfaces stuck one to another, we actually will have a neurological symptom of that, and that's actually pain. And so people with pericarditis most commonly will have chest pain associated with that pericardial sac being stuck and actually moving with the heart itself, and it's not designed to do that. The other thing that I want to point out for you guys with the pericardial sac is that it is not free. With our other specimens that are quadrupeds, the pericardium around the heart doesn't have really any surface attachments to other structures other than the base of the heart. However, in humans, because we're bipeds, our pericardial sac, if you'll notice, is attached to the diaphragm. And so we have truly a diaphragmatic surface of our heart, and the reason for that is because that pericardial sac is attached to the diaphragm. Now, this is a very good way of keeping the heart in its space, correct? So this works for us very beneficially, especially since we are bipeds. It keeps the heart nicely centered in the mediastinum. So the mediastinum is that space between our two lung fields, and it has an upper aspect, which we're not going to go over, but in the lower half of the mediastinum, that's where our heart and pericardial cavity and sac is located. Now let's take a look at the layers of the heart wall itself. We have the epicardium, which we also call the visceral pericardium. We have the myocardium. And don't let the notes confuse you. It says fibrous connective tissue, skeleton plus cardiac muscle. Well, when you look at the myocardium, it all looks like muscle. Because remember, the connective tissue, quote, skeleton is laden with all these muscle cells, and so that's actually all you appreciate. But you have to realize that that CT is there, otherwise those cardiomyocytes could literally float away from one another without some sort of scaffold to be attached to. Endocardium lines the chambers, so it lines our atria and our ventricles and also covers the valves. And this endocardium is simple squamous epithelium. So simple squamous epithelium is going to line everything inside the heart, even our chordae tendineae and our papillary muscles, all that stuff is going to have a nice little simple squamous epithelial lining all over those. And we're going to find out that also inside our blood vessels, it's simple squamous epithelium that's lining our blood vessels, whether they're arteries, veins, or capillaries. And so our blood, which is enclosed in a closed system, only actually sees simple squamous epithelium throughout its transit through the circulatory tree. Now let's get into the part of the notes that says chambers and great vessels. The heart has 4 chambers. 2 of them are the same, 2 atria and 2 ventricles, and it has 2 types of vessels. And so this is where it's real important that we make sure we affix this definition of blood vessels in our mind. Blood vessels, arteries. Arteries are blood vessels that take blood away from the heart. And veins are blood vessels that take blood back to the heart. If you're a vein taking blood back to the heart, which chamber does that blood go into? Goes into the atria. So remember, for our heart, veins are always emptying their blood into our atria. And therefore, arteries are always emanating or originating from our ventricles, because ventricles are the pumping chambers responsible for taking blood away from the heart. This definition has to be in place because you cannot think of arteries as always carrying oxygenated blood and veins always carrying deoxygenated blood, because we have the pulmonary trunk, which is an artery coming off of the right ventricle that is splitting into right and left pulmonary arteries going to the right lung field and the left lung field for oxygenation. And so that artery is actually carrying deoxygenated blood. And so that's one of the things that you guys have to realize when you take a look at what I call the cartoon characters in your books that shows you red and blue for what's inside the vessel. Realize that red is referring to oxygenated blood and blue is referring to deoxygenated blood. And so you will find blue blood in an artery. But the only arteries that are going to be associated with that type of changeover of what type of blood we have in arteries versus veins is actually the pulmonary circuit. And we'll go into some more detail on that in just a little bit. Chambers is our next topic here. And 2 upper and 2 lower, we consider the atria the upper chambers and the ventricles the lower chambers. The bulk of the heart, however, is ventricular. So the mass of the heart is ventricular. Our atria indeed possess thin walls because they are designed to be low pressure receiving chambers. They are designed for distensibility. So they can balloon up a little bit for you. When we take a look at the parts that all of the atria have, both atria have auricles. And auricles, just like what we were doing with our special senses, auricles are ears, and they are anterior ear-like flaps that actually have within them these next item on our list here, pectinate muscles. So pectinate muscles are in auricles. So you can have an arrow to pectinate muscles and from pectinate muscles back to auricles. And that is the muscle appearance, the bundling of the muscle appearance that are actually inside the auricles. What we have with these auricles, they are actually the only muscular, what I consider muscular aspect of our atrium, or atria for plural. So that's the only really truly muscular aspect of those. And they do allow for a little additional blood volume. So they do provide a little more space for blood volume. And because in the auricles, these pectinate muscles are located, it's interesting if you actually take a look at the linear arrangement of those pectinate muscles, because they will actually cause those little atria to shorten. And so that's how that blood is actually pushed into the remainder of the atria themselves. And so when we talk about atrial contraction, we're actually talking about auricular contraction. Are you guys following me on that? Because that's the only truly muscular area for that. The fossa ovalis is also in both atria because it's found as an oval depression in the interatrial septum because it is a remnant of a foramen. So the fossa is a remnant of a foramen. And remember, a foramen was a hole, and it was called the foramen ovale. So the fossa ovalis is a remnant of the foramen ovale, which was a shunt, which was a fetal shunt. Because we don't need a bunch of blood going to our lungs for oxygenation when we're in utero. The blood goes to the placenta for oxygenation. So we don't send the usual volume of blood to fetal lungs, which are only at that time actually just developing. So fetal lungs only need enough blood going to them to actually promote their growth and development. You don't need the bulk going to that for oxygenation. So the fossa is actually a remnant of a hole that got covered up after the baby was born and becomes an infant. So it actually closes off fairly quickly within the first few weeks to months of life. So that's what we actually find there. And I want you to realize it's in the posterior aspect of the interatrial septum. So found in posterior interatrial septum. And where you find actually the coronary sinus emptying into the right atrium, you will be able to more easily find the fossa ovalis in that area. And since it's between the two, you actually have it identifiable on either side. So when you do your heart eval, make sure you mention that whenever you're in the atria, because that's part of your identification. So let's take a look at the part of the notes that talks about some of the differences between the atria, the right atrium and the left atrium. Well, the right atrium is receiving used blood from the entire body. So that used means oxygen poor, carbon dioxide rich. And three ways to get into the right atrium are the superior vena cava, the inferior vena cava, and the coronary sinus. The superior vena cava is bringing used blood from areas above the heart. And the inferior vena cava is bringing used blood from areas below the heart. And the coronary sinus is actually bringing used blood from the heart muscle itself. So the heart, even though it's full of blood, does not use that blood. It's not designed for it to use because the heart is designed to pump the blood. And so in order to feed the heart, we actually have its own circulatory system, which is called coronary circulation. When we take a look at the left atrium, the things that differ from the left and the right is that pulmonary veins enter the heart, and this is bringing fresh blood from the lungs. And so that means with fresh blood, that's oxygen-rich, carbon dioxide poor. So right atrium, used blood, left atrium, fresh blood. So therefore, that blood is now going to descend into the ventricles, which are the actual pumps. So let's take a look at some of the things associated with our ventricles. Our notes say here, relatively thick walls. Absolutely. When you compare them to the atria, the walls of the ventricles are incredibly thick, both right or left. However, one is definitely significantly thicker than the other, and that's the left ventricle. So the left ventricle is significantly thicker than the right ventricle, and that is because the left ventricle is responsible for pushing blood out into the system. So your entire body except for your lungs, because the right ventricle is responsible for sending blood to the lungs. So the right side is often referred to as your pulmonic circuit, and the left side is referred to as your systemic circuit. We'll get into that part of the notes in just a moment. Parts that are in our notes itself say the interventricular septum. Well, absolutely. We have a wall between the two ventricles, and its thickness is more like the left ventricle. Inside, you see a very definite ridging pattern on the inner aspect of the walls, and they call that trabeculae carneae. Trabeculated is kind of like a lattice or lace-like networking appearance to that. Then we also find inside our ventricles these muscular projections called papillary muscles. Our notes say small stalks of muscle extending from the wall. So you'll find them on the right side, you'll find them on the left side. These papillary muscles, if you look down to our next topic, of which will be heart valves, are actually anchored to the atrioventricular valves via chordae tendineae. Your book also calls them tendinous cords, and that's another name that you can use when you do your eval, but we're going to call them chordae tendineae here because that's what we've been calling them for decades, and it's a name that still persists out there in the healthcare field. So the papillary muscles are the attachments of chordae tendineae to your heart valves between your atria and ventricle. It's very specific as to where you actually find those. Papillary muscles can contract, and we'll talk about why they contract in just a moment, and when they contract, you will not see them protruding upwards out of the myocardium, the heart muscle itself. So if you see chordae tendineae without any papillary muscles, you have to realize that the papillary muscle was actually in a contraction mode. If you see papillary muscles, that means it's partially contracted or relaxed based upon the length of the papillary muscle itself. Things about the right ventricle that are a little bit different from the left is indeed, our notes say, relatively thin wall. The other thing about the right ventricle is that it has a pulmonary trunk. Responsible for ejecting the blood out of the right ventricle into the pulmonary circuit or towards the lungs. So pulmonary trunk sends used blood to lungs for oxygenation. The left ventricle has thick walls because it's sending its blood throughout the entire system, whereas the right ventricle is only sending its blood to its next door neighbors. So based on that, the left ventricle has to generate more pressure to get the blood to go further away. The right ventricle doesn't have to generate as much pressure to send the blood away because it's literally right next door in this actually vacuum-like cavity. So for notes, left ventricle, relatively thick walls. The aorta is the blood vessel that takes blood away from the heart or the left ventricle to all of the body except lungs. So takes fresh blood to body except lungs. The other thing that I want to talk to you for a moment right now about the right and left ventricle, after you've already taken a look at them, you've already seen the inside chambers of the right and left side, and you've learned that we actually have different appearance to the atrioventricular valves, and we have different names for them. I want to ask you, I want you to realize when the heart ejects blood, both ventricles are ejecting blood at the same time. So whenever we have a heartbeat and we have what's called stroke volume, we'll get into more details about that. When that leaves the heart, this is always happening at the same time, and the reason you hear heart sounds are actually due to the closures of the heart valves. I want to ask you, do you think the right side or the left side pumps more blood, or do you think that they pump the same volume of blood? Let me ask you this question. For those of you that think it's going to be difference in volume of blood, remember we're a closed circuit, okay? If the left side is pumping more blood than the right side, what happens to the filling of the left side ultimately? Because where is its blood coming from? Isn't it coming from the lungs? And the right side is sending blood to the lungs? So you have to realize the right and the left side are actually pumping an equal volume, because if you weren't pumping an equal volume, you're eventually going to be shorted before you can actually start to pump, correct? So you have to realize that even it's an amazing thing to think about how much blood is actually being pumped to the lungs. The same volume of blood is going to the lungs as going to the body. So your lungs are really able to hold a large volume of blood themselves, and that's why your lungs, whenever you actually get a hold of real lung tissue of a recently butchered animal, it's just like sponges. So your lungs are like sponges, and you guys know how much fluid a sponge can hold. And so your lungs hold a lot more fluid and blood than you actually realize. They hold a large volume of blood themselves. So right and left side must pump the same volume, because the left side is dependent upon the right side filling it up. You guys follow me? And vice versa. So this is a circuit, and if you short any side, it will affect the other side. So you always have the same volume of blood. Now let's take a look at the heart valves. The heart valves are placed in two areas. They're between the atria and the ventricle, and they're at the exit of the arteries from the ventricles. We call the heart valves between the atria and ventricles AV valves, and as our notes state, these valves are absolutely responsible for keeping blood flowing in one direction, which is from the atria into the ventricles and out. The AV valves are the tension on the AV valves is adjusted by the next two items, the chordae tendineae and the papillary muscles. In the pictures below, one says atrioventricular valves open and the other says atrioventricular valves closed. So this doesn't matter whether we're on the right or the left side. You can tell on the first picture that you're actually taking a look in ventricles, because take a look at the texture of the wall. That's trabeculae carneae. So that's an example of trabeculae carneae in our cartoon here. The papillary muscles are easy to identify. They're projecting up. You can see the notes say tendinous cords, but we'll call them chordae tendineae, attached to the papillary muscles and also attached to the valve leaflets or cusps. I have a tendency to call them leaflets, but also they're called cusps. Blood is going to flow from the atrium over these valves, which should be essentially flat against the wall of the ventricle, because that's where they're going to be pulled down into, is into the ventricular wall. So when blood flows in, these valves are open. And then when the ventricles contract, in order for the blood not to squirt back into the atria, we need these valves to close. One of the things that's difficult when you see these pictures and you see the hearts, you want to think of the heart as actually contracting, like if you had your heel of your hands together and snap them together. That's not how the heart muscle contracts. The heart muscle acts a little bit more like if my hand flat up here are the area where the AV valves are located, and my fist here is the apex of the ventricle. It literally starts to go up towards the AV valves. So the heart muscle actually twists and shortens as the ventricles contract and eject their blood out. So the blood and the muscle is coming up towards the AV valves. You guys following me with this? So the papillary muscles are there to actually contract when the ventricles are contracting, because as the muscles, the papillary muscles and their chordae tendineae and valves come up towards that hole, if we don't start to shore up the tension on the chordae tendineae, those cusps or leaflets will flap into the atria. Are you guys following me with that? And if they flap into the atria, we get regurgitation. So we get regurgitation of blood or prolapse of blood into our atria. So the valves themselves, that action where they are flipping up into the chamber that they're not supposed to flip up into, which are the atria, is called prolapse. And when that happens, of course, then you'll have backwards squirting of some blood, and that's also referred to as regurgitation. There are several people out there with heart conditions where they have mitral valve prolapse or tricuspid valve prolapse, and that's basically telling you there's a problem with the tension on the chordae tendineae, and your tension on the chordae tendineae could be a dysfunctional papillary muscle, correct? So a dysfunctional papillary muscle could be part of a prolapse, or what if you had busted chordae tendineae? If you had busted chordae tendineae, what will happen to that leaflet? It's flapping in the breeze like a sail on a sailboat that doesn't have the proper tension anymore on it. And so you end up getting blood that goes backwards or regurgitation. So this arrangement is actually very important to actually keeping blood from flowing into the atria. As long as everything's working properly, you don't have to worry about that. So that's the atrioventricular valves. Now on the right side, take a look below the picture. We have tricuspid is the name for the AV valves on the right. And for the AV valves on the left, we call them bicuspid or mitral valves. So tri means 3 cusps or leaflets, and bicuspid valve, 2 cusps or leaflets for that. One of the reasons that it's speculated that we have 3 on the right side is that the inner dimension of that chamber is more irregular, and so we actually need to actually fill in that irregularity with an additional cusp. Semilunar valves are the next topic for valves here. Notes say at the entrance of aorta and pulmonary trunk. So that means where the right ventricle is now sending its blood into the pulmonary trunk, it's right there in that area. Same thing for the aorta. These valves are completely different. We have no chordae and no papillary muscles associated with them. They are called semilunar because when you take a look at them, you see a bit of a half moon. And this is what's important about them. They're more like pockets in their construction. And this interesting pocket construction also helps to prevent prolapse. In the way to think about the semilunar valves, there are 3 on each side. There are 3 in the pulmonic trunk, and there's 3 at the base of the aorta. The difference with the aorta is that we have little holes in 2 of the 3 for coronary ostia for right and left coronary arteries. But I want you to think about the semilunar valves like pockets in the aspect that they are flat against the wall when blood is being ejected out, and then when blood comes back, the blood itself fills up the pockets, causing the pockets to come together in the midline and snap open. And it's just like if my legs were ventricles and they were pushing blood upwards out through the aorta, for example, and it makes sure that the semilunar valves are truly flat against the inner wall of the aorta. Then as the blood does what blood wants to do, once you've squirted it out, where does it want to go? It wants to go back down. And for many different reasons, but let's just pretend we're just going to only use gravity for right now. That's why I like to use the up and down thing. So the blood wants to come back down after it's being squirted out, and it fills the pockets and distends the pockets. And these semilunar valves are designed, because if you ever take a look at a human one, they have a little bitty, almost a little bitty dot on the mid of the semilunar valves where they all come together. So they're all designed to snap open, all 3 of them, and close off the entry or exit way into the ventricle from that side. And so you have to realize that the blood flows up and bounces down almost on those semilunar valves, causing them to snap shut. And when it does so, then the blood bounces again and bounces on its merry way down the aorta or the pulmonary trunk. And we'll discuss a little bit more of that mechanism when we get into that. But I want you to realize semilunar construction is very effective, but it is completely different from the AV valve construction. Our notes about aortic semilunars have a little space to actually write into that. And so we have 3 cusps. The aortic semilunars contain ostia in 2 of the 3. And the ostia are the beginning or entry portal for the blood flow to the heart itself. And of course, aortic valves control flow of blood into the aorta. Now, well, that's kind of self-evident, but whenever there is certain things like stenosis, stenotic aortic valves will greatly impact the flow of blood into the aorta, because stenosis means narrowed, correct? And so that means that we'll get less blood actually flowing into the aorta if we actually have certain things like aortic stenosis. Also, we'll have incomplete closure of those valves too because of that scarred up situation. Pulmonary semilunars, again, have 3 cusps. Control the flow of blood into the pulmonary trunk. And that brings us to coronary circulation, which is what you're supposed to be finishing up this week with your heart muscle dissection so that you get it understood and can follow it out. Flow of blood to the heart has its own separate circulatory system or unit, and that's called coronary circulation. So when somebody has a heart attack, we often say they are having a coronary. So coronary is different. Right now, when you hear coronary circulation, that's different from cardiac circulation. I want you to realize coronary circulation is referring to the blood flow to and from the heart muscle itself. Cardiac circulation is going to more or less refer to the flow of blood through the heart muscle itself and what circuit it's going to be going to, whether it's the left side and systemic or the right side and it's pulmonic. take a look at your coronary arteries. This is actually a really good picture. It's much better than our previous text as far as the picture of our coronary arteries. Our notes say they exit the aorta via coronary ostia. So ostia is a little hole. Those, again, are located inside 2 of the 3 semilunar valves, and so you have to realize you have to look inside the semilunars to actually find these. The coronary arteries will eventually become capillaries. They'll get smaller and smaller, and the capillaries are actually going to feed the heart muscle itself, and then the used blood is going to go back to the heart and eventually into the coronary sinus, and we'll get to that in just a moment. Take a look at your picture at the right coronary artery. I want you to realize it's going to emerge kind of from behind the pulmonary trunk, because our aorta is kind of behind the pulmonary trunk, and you'll find it in that groove or sulcus between the atrium and the ventricle on that side, on the anterior aspect. That is more an auricle than it is really an atrium, because the auricles are just the anterior manifestation of our atria. Our atria are predominantly posterior structures. So we have a nice little groove that the right coronary artery is going to be found in. So that's a groove or a sulcus, atrioventricular sulcus on the right. And what you want to do is you want to dissect that out, and you want to see if you can find the right marginal artery, which is on the inferior margin of the heart. As we follow the right coronary artery around the backside, it will give rise to, in the majority of individuals, to the posterior interventricular coronary artery. Now, you guys will have cut through this artery a couple of times, because we have quite a few. We have at least 2 posterior cuts on your heart. So you have to realize you actually will have cut through this a couple of times, but you still can follow it and find it in the posterior interventricular sulcus. So make sure you dissect out enough of that coronary artery to demonstrate that, and put your probe underneath those things. That's the right side. Now let's take a look at the left coronary artery, and what I don't like about this one is that they make the left coronary artery look, how long would you guys say it looks like the left coronary artery looks to you in that picture? A couple of inches? Yeah. It's about 1 centimeter or less. Okay, so that picture is designed to show you where it goes, and it isn't well designed to show you its length. Okay, so again, this is going to emerge from behind the pulmonary trunk, from our aorta, and it's going to be very short. It's going to be 1 centimeter or shorter on your specimen. And then it's going to branch or bifurcate. And that branch will give you 2 main vessels, the anterior interventricular coronary artery and the circumflex coronary artery. And this picture doesn't do a very good job of showing you the circumflex like I'd like it, but anterior interventricular coronary artery is on the anterior interventricular sulcus. It's easy enough to get these guys dissected out when you put your blunt probe into the coronary ostia, inside the aorta, and then look on the outside, and then wiggle it around, and then you start to remove the fat around those blood vessels. You also will trench out some of the myocardium to get down deeper to that, and then you'll be able to use your blunt probe to actually get underneath it and follow it on out. So the anterior interventricular coronary artery is on the obviously anterior surface, and the circumflex coronary artery is in the left atrioventricular sulcus or groove. So remember on the right where we found the right coronary artery in between the right atrioventricular sulcus or groove, the circumflex, and circumflex means circumferential, so it's circling around. The circumflex is in the left atrioventricular sulcus or groove. I notice on these pig hearts they actually have an additional one at this bifurcation. They have a trifurcation. They actually have 3 branches on most of the specimens that I've looked at. And I would call the one in the middle the left anterior ventricular coronary artery, because that's where it's going to. So you have to realize these vessels, and as we get into naming of vessels, they're typically named for where you find them, or if it's an artery, where it's going to, or if it's a vein, where it's coming from. Looking down now at the coronary veins, there are 3 coronary veins you're supposed to identify. You're supposed to identify the great cardiac vein, which is going to be on the anterior interventricular sulcus with the anterior interventricular coronary artery. And I notice it's a little bit to the left of that artery. And it's going to go up and it's going to go over the circumflex artery. One of the things about arteries to remember, they are muscular and veins are flabby. So if you always remember arteries are muscular and veins are flabby, you'll understand why the veins are typically on the surface of arteries versus the arteries being on the surface of the veins. Arteries are high pressure systems that actually have muscle in their walls to help them squeeze the blood to wherever they need to go. Veins are like atria. So veins are actually designed for passive collection. And if you're a passive system and you have something strong sitting on top of you, you will basically be applying a tourniquet to those veins. So you have to realize that veins typically will always want to actually travel over the surface of the artery, if not just adjacent to the artery. And that's what our great cardiac vein does. The middle cardiac vein is going to be found in the posterior interventricular sulcus, adjacent to the posterior interventricular coronary artery, and the small cardiac vein is going to be found with the right marginal artery. And so that's how you actually work about finding them. They all empty into the coronary sinus, and remember the coronary sinus is a posterior structure. So it's between the atria and the ventricles on the posterior side, and empties into your right atrium. So it's taking the used blood back to the heart. The only thing I would add to the notes on coronary veins, blood from the 3 main veins are returned to the coronary sinus and then into the left atrium. Then right atrium. I may have said left, didn't I? Yes, right atrium. Going down to the pathway of blood flow through the heart, 2 main circuits. The pulmonary circuit means that blood is going to the lungs, and the systemic circuit means that blood is actually going to the system or the body. The pulmonary circuit, its function is to reoxygenate blood. Function. And it is often called the right circuit because it's essentially from the right side of the heart. Systemic circuit is going to take fresh blood from the heart to all parts of the body except for the lungs, and then back to the heart again. And this is often referred to as the left circuit. We have 2 diagrams below, one showing you a little bit about the pulmonary circuit and the other one showing you about the systemic circuit. But one of the things that we want to do real quick is do a little drawing about the 2 sides. And I want you guys to work with my words and make your own visual drawings. In the center of that big space under all of that, I want you to make what looks like maybe a bell or a cup. And inside, I want you to make 2, I want to call them a lung on each side. I cannot find a piece of paper. Anybody got a piece of paper that I can borrow? Or not borrow, actually have. I'll draw it in 2 places. Put this on a pause. Okay, so let's go ahead and draw some more to this diagram. So right here we've drawn our thoracic cavity and lungs inside. Now I'm going to go ahead and draw a rectangle off to one side and another rectangle off to another side. And I'm going to partition my rectangle to have a smaller upper space and a larger lower space. And I'm going to call the top part of my rectangles A for atrium and the bottom part of my rectangles V for ventricles. And now I'm going to draw two blood vessels from one atrium connecting our lungs and one blood vessel connecting our lungs to the ventricle on the other side. So my question to you is, which side is the right and which side is the left? Based upon this picture, this is the left side and this is the right side, because these vessels right here are pulmonary veins, so PVs. And this vessel right here is PT, or pulmonary trunk. So now to actually finish these circuits up for us, we know from the left ventricle, the artery leaving that is our aorta. And it takes blood to our body. So this is our body here. And then blood, fresh blood to our body, and then used blood from our body will go to the right atrium via two main vessels. This one here, and then this one here. I'm just going to cross over another path. And so you should realize that this is meant to be the IVC, and this one here is meant to be the SVC, or superior vena cava. Now on the left side, the atrioventricular valve is going to be the mitral valve or bicuspid valve, and on the right side, the atrioventricular valve is going to be the tricuspid valve. And so what we want to do now is I want you to think about what would happen to the flow of blood if we actually had dysfunction of either our bicuspid or tricuspid valve. And we'll talk about that a little bit more when we get together next time.